So, good morning, everyone. This is our first, I hope, the first but not last scientific cafe.
You know, just don't announce anything.
Yeah. We had a very long history of Grand Office Cafe, where was great service in cafe, croissants, and everything. And we discussed the project-related stuff. And we really enjoyed the time, and we said maybe there might be a potential to go farther and not to talk only about how to write a project, what are the actual calls, and who to contact with the admin staff, etc., and start to actually speak about science here, the Faculty of Medicine. So we want to try. The croissants are here, coffee is here, and we have special guests here as well, with whom we will talk about life in science. And my invitation accepted Aleš Hampl from the Department of Histology and Embryology, and Bárta, head of his research group, Bárta research group, from the same department. So the concept will be, we will have a discussion about the career, if there are some general stuff that we can extract from living experience that you who are a bit younger than Aleš and me could use for your careers as well. And then we will touch on current grant call of the Gotcher. So there will be a part dedicated to that. And finally, the project office got some news about gender in science. So we will ask Jitka Blašková to come in and present what Gabina and she and their team prepared for your applications. So, good morning.
Good morning.
Again.
Thanks for having us here.
This was a long introduction, but we have a lot of time. I said 1 hour is something like a typical psychiatric interview, so I know that we can go into deep details. We got a lot of time, and I will start with Aleš. Aleš, you have a lot of things ahead of you in research and science, because you are dedicated head of the preclinical center that Masaryk University is working on to be here. But you have also a lot of behind you already. So you were the Vice Dean for Research under the Dean Professor Meyer, and you kind of made the Department of Histology a kind of Academy of Sciences department. So I would like to ask you what brought you here. What is your vision that you started from scratch, and now you are sitting here as a guru of science here. That's cool.
I don't know if I am any guru at all. First of all, I have to say this experiment on me and Tomáš has never been approved by the ethical committee of the Faculty of Medicine. Right. Anything will happen.
I fully agree with that.
To any of us.
No. This is covered by health insurance. Yes, so this is a different.
Then I feel much better, actually.
Yeah.
All right. Yeah, my career started really several decades ago. I'm older than I look. I started my university in the era before the Velvet Revolution, so imaginably the career started as a helper to my boss at the Department of Histology. It is something that none of you actually know, that I was a histologist 30 years before I became head of the Department of Histology and Embryology. So my career started as a morphology researcher. Mm-hmm. But then, for a major luck to me, I was not given a position at the Department of the Histology and Embryology at the veterinary university. As if that would have been actually happen, I would probably be staying as a assistant professor till the end of my life. So fortunately, it did not happen, and I got an opportunity to do my PhD, and not really a PhD. I did it with a candidate of science degree at the Institute of Academy of Sciences, as this detached laboratory in Brno. And then there was another shock to me that my supervisor died, and my real supervisor became head of the institute, who I had a chance to see twice a year when he came to Brno. And my only duty at his visit was to take care of an ashtray for him to smoke one Camel cigarette and leave. So that was pretty much how my PhD went on. But then there was the third luck in my life, career life. That was when Peter Dvorak came to Brno. And Peter Dvorak, at these days, was one of the pioneers of hybridoma technology, which is the technology that leads to production of monoclonal antibodies, as you all know. And this is what changed, upside down my development in research. As from Peter, the majority of what I learned in terms of cell culture and cell biology is from Peter. That happened in years 89 to 91, when I defended my candidate in science degree, and half a year after I left for the United States, where I spent as a postdoc at the Jackson Laboratory 4 more years. And then I got back to Czech Republic, and there was one question in the list asking me what I did to expand my group. Actually, I did nothing, as there was, the situation was completely different what we experience these days. I was not really thinking about what will happen after coming back. I wrote a grant to Gotcher, these early Gotcher grants. I got a couple hundred crowns, and I moved in here to learn what the situation is. There was no connection to Brno for the last 4 years. I had no idea how the thing looked like. So I was pretty much shocked that I had some money. I was in the institution, but there was nothing around. So this really brought me into a situation that I really had to invest quite a bit of energy to establishing everything I needed for my work. So it was not a real science. It was not sitting and thinking about experiment. It was like trying to find the basics to get the pipettes and the tips. So there is nothing compared to it currently. Nobody is exposed to that situation. Again, luckily for me, Peter got back from Japan and France, so we rejoined after several years, and being 2 of us, having a little more energy together, and the support from one side to another one, then we succeeded and moved from one institution to another one. We've been hired by Eva Sikova, who was heading the Institute of Experimental Medicine at these days, and she hired us because of I guess one of the major, I don't want to say discoveries, by breakthroughs in our research. It was establishment of the first lines of the human embryonic stem cells, and that catapulted both of us really, tremendously to the clouds, as now we were attractive to international community as one of the groups around the world that succeeded in this task, and this completely changed our lives. There is one more thing that I want to stress. There was an important person in my career, and there's Professor Jan Motlik, who was the one who told me and Peter, you guys, what you should do is to start investigating early embryos and embryonic stem cell lines. And he told us this, we did not really know what these cells are, so we learned that it helped quite a bit. And then, you know, there was stepwise process. We moved to the Faculty of Medicine, thanks to Professor Iri Meyer in 2005, colonized the new pavilions, and then we succeeded in the fight for the position at the Department of Histology, and then almost all my young colleagues came into action.
Amazing experiences. So you got something like an analog of PhD here, candidate of sciences, and then you moved to the US for 4 years postdoc, and then you came back here and you started from scratch, building from scratch on the green.
Pretty much. You know, as said, I wrote the Gotcha project back in the States, and I got the money. There was a project on animals as I did my postdoc at the Jackson Laboratory. It was
what did you do there?
There was gamete development on mice. So the Jackson Laboratory is premier research center in the United States for mouse genetics. So I had a project on mouse genetics, and I came to burnout, and the head of the institute told me, it's nice that you have project on mice, but we don't really are interested in supporting mice research anymore. So it put me really into the hard situation, but even though we made it somehow.
I just try to imagine how is it that you come from the Jacksons and the let's say infrastructure and processes supporting research are there, and you came here early in the 90s back, and there's almost nothing. So how did you, let's say, capitalize on your US experience to work here as a researcher? Was it anyhow, let's say, something like a vision, what you have to build around you to do the research, or was it, there must be also some, let's say, disillusions when you when you worked for 4 years in a research institution. Now you came back here.
Well, I think that's the advantage of my personality, that I'm pretty stubborn. And I'm not really affected by disillusions. I always think there is some positives in future, and it's only a matter of time when it will come. So we managed. And there was a beauty of being in the States these days with no connection to Czech Republic besides the mail and phone. So I had no idea what's in here. So we just got back and see.
This is really a pioneer experience. I wanted to ask you about what you could use from the US experience, but my personal experience is from the stay abroad was I really need this and this and that to be able to promote my research. Because I was in a clinical setting and there was completely nothing out in terms of research infrastructure and research support. It was all dedicated to pharmaco studies, etc. But something for neuroscience, there was zero. So I imagine that you came back here to a similar situation.
I don't think the situation in the Academy of Sciences was that bad these days. There was an institute. There were some instruments. There were even the animals. So everything was just, be patient, collect the materials. Okay. It takes time, and find people who will share the dedication to science with you.
Another important point that you wanted to stress is that that your professor or supervisor said you should go this direction because there is a potential and focus on embryonic stem cells. So this is something that really helped to get to the international level. Yeah, to be somewhere where it's, let's say, on the edge. You should, your supervisor knew there's a big potential, and the technology is already there to promote the kind of research. Yeah, so this was an important
There
part.
was no technology these days that would suggest that embryonic stem cell will be a good thing to study in future, but for some reason he felt that this could happen actually. The Jan Motlik's, the history of Jan Motlik is not an embryonic stem cell, but it's reproductive biology. So he was an expert in oocyte development. And this was actually to where he pushed me, helping me to find a position in the States. And the supervisor that I had in the States, Dr. John Epic, was friend of Jan. So that was the way how I really, it took me just minimum time to find the position there.
So the role of the supervisor for early career researchers is to promote networking and to find
very
a niche
very
where to go. Yeah.
Yes, it is.
Yeah. So if you compare your experience of starting your independent research and a situation of someone these days who wants to start, what's the difference? What's the same? What's the continuous theme in deciding to be a PI and promoting a research career in my life?
It's a combination of two things. Decision to do something that is interesting and it's nice to do, and to be lucky and find the proper thing that is interesting. It's promising for future research and having it as a direction for long term and not only for 1 year, from one grant to another one. There's one actually property that I brought with me, which is dated 1994. And this is what's been copied by my American mentor. And this has been published in the New England Journal of Medicine as a commentary. It's 4 pages. I wanted to copy it and distribute it in here. And it's named Picking a Research Problem, Sounding Board, Critical Decision. You read it, and you know everything what you should do. It's on 4 pages, and I have certain.
We will scan it and
I have some of these elements underlined. Like I can read. Pick areas that are not only of interest to you, but also of interest to a large fraction of the scientific community. Then another one. Be prepared to sell your ideas. Become the locally recognized expert in some area, which is definitely very important. Many others.
Yeah, but with the first thing, I wouldn't agree, because you might end up in the field that is super highly competitive. It's rather to pick something that you are really passionate in and really want to address, or you can pick, I don't know, some really unique model to address unique disease or whatever, or specific signaling pathway or whatever. So I wouldn't say these days it's really true to pick something that is really super interesting for everyone. These days, it might be you ended up in highly competitive field, and sometimes
but
maybe
that's
you may
also
suffer.
in here.
I don't know. That's
I could cite everything that's written. The project should have the potential to yield a seminal observation.
That's definitely true.
Yeah, and I think that you need a long-term vision, because everyone can see that science goes in let's say circles, and everything, fancy stuff appears that everyone's doing this, and then it's going nowhere. So another fancy stuff, and everyone's doing that again. So if you have a passionate topic and you keep your track, so I think
if you are so lucky to find your, let's say, niche, that's the best way. I think in these days.
Yeah.
There's a question.
Yeah, maybe I have questions, or is there...
Definitely, yeah.
So I have a question for you, Dr. Bárta. So you're saying that to go to the really competitive area for early investigator is not the best choice from your point of view. Let me finish. So, thank you. So shall I repeat it so everyone hears it? So going to some, let's say, broad but competitive area of research for early career investigator is one choice, or finding your own niche, really specialized, is another choice, and all those possibilities in between. So what would you say about, let's say, painting the picture of your own niche to make it really interesting for the grant reviewers? Because ultimately it comes down to, okay, I've got this really exciting problem I want to study, and I'm really passionate about that. But how to make it really interesting for someone who has no idea, no clue what you're talking about, because it's a grant reviewer assigned to your proposal who really never heard about that thing. So how to strike the balance?
That's very brilliant question. For me, my major model are retinal organoids, so small retinas or small eyes, let's say human eyes, and there's a very big competitive field in terms of retinitis pigmentosa and other vision diseases, let's say, that affects a huge number of people in the population. But I focus rather on super rare diseases. So if you pick super rare diseases that no one is that interested, but they are interested from point of view from basic science, because you always look at the one specific gene on one specific mutation in a gene, and then you figure out maybe even bigger picture how the things work. So you are not studying disease that there's hundreds of papers every month, for instance, but you focus only on specific disease or specific mutation, and you can pick several projects on different genes, different mutations, but these are super, it paid for me. It's not general advice to follow these, but it's really something for me. It's really driving force to pick something really interesting and weird and to address this.
So if I got it right, so you're saying to compensate for really narrow niche would be to make it interesting on different layers, let's say from technology point of view, the model point of view, the basic science point of view, so you can reach maybe not the breadth of various, let's say, a field which is interesting to everyone, but looking from different directions that the possible reviewer can find something cool for himself, maybe not as like the primary focus of the research, but let's say the technology or all these things you mentioned. Did I get it right?
Yes. I always bid into basic science rather than applications, so I always focus my research on basic science. So yeah, you're right, as you say, focus and try to convince the reviewers that your idea and you have the instrumentation. And for the reviewers, always it's good to point the general hypothesis of your project and test this hypothesis multiple times using multiple approaches, potentially multiple models if you are capable of doing multiple techniques, and it always sounds really good. And if one approach failed, there's always a number of different approaches to address the general hypothesis. The general hypothesis doesn't have to be, I would say, sexy or in major interest, but it's really solidly built, and you test it multiple times using many approaches. It always sounds good. If I were the reviewer, I would think so.
Great, thank you.
Yeah. So another point of view is that even though that you study a very narrow niche, you can extract a general biological process out of that, something that is more general, and this is just an, let's say, extreme, extreme way how the general process is expressing itself. Yeah.
Exactly. For instance, we have one family in Czech Republic having single point mutation in one microRNA and they are blind. No one knows why. And this is super rare disease. There's another family in the US, I've been told. So there's a super unique model. We can address the mechanism because no one knows what's behind this. So this is something that really may reveal a new molecular mechanisms. We will not treat the disease, but we will understand the mechanisms behind this, let's say, phenotype.
Perfect. And this is also a bridge, but we have another question over there.
Thank you very much. Actually, I don't intend to ask a specific question, but I just wanted to share a little experience of mine that I think would resonate with the discussion that is going on. First of all, my name is Yusuf Surakat, and this is my very first week in Borneo, and I'm very much happy to be here. I'm pursuing a PhD in public health, and so the little contribution I actually had is about the issue of choosing a niche of study. So my first degree was in human medicine, and I practiced in the clinic for some time, but my passion had always been working in the community. And as such, my interest was to work in the areas of tuberculosis. However, tuberculosis has been saturated in the sense that there are so many researchers globally that are working on tuberculosis. Nevertheless, because of the fact that that's where a lot of resources are and that's where I could get the knowledge, I enrolled in certain programs that were anchored by WHO, USA, GLRA, and the likes, and I gained some knowledge. Come 2019, and then COVID-19 comes in. It's a new area, so not so many papers have been written about COVID-19. There are so many things to research. Now, if I'm able to identify a niche, and then I become a professional, a specialist in that area, coming back to his question about making it interesting to others, I believe that if you've mastered a topic, you've mastered an area, you've researched, you've published, you've interacted with people, other people who research in similar areas, I believe you will be able to make it very interesting for the reviewers, for the listeners, and overall, your passion and your contribution to knowledge supersedes everything else. Thank you very much.
Thank you for your comment. We moved a bit to the Gutcher part of the so you said what do you
I said what worked for me. It's
what
not general
works for
advice.
you.
Maybe someone will find niche in super interesting, widely published topic. I'm not saying that it is not right. It's just what worked for me.
Yeah, definitely. And it really depends on who you are, what's your career track, what's your network, etc., etc. Yeah, but you said an important point. You have a sound hypothesis and many models how to test that. Yeah, so you go pretty deeply and you have a let's say extensive way how to test your hypothesis. Yeah. That's what works for you. Yeah.
Not the only tip. The second tip is really to have a good collaborations, because you are not able to do really good projects by your own. Especially if you are a young investigator and have a small team, you really need somebody who will do at least some parts or some critical experiments that you are not expert in, or if some of your collaborators has some specific unique model, you can always reach them out and ask to collaborate. Or at least there can be just letter of intent that they will be collaborating with you. They don't have to be co-applicants of your Gutcher application. So just this letter of intent always helped me. For instance, because I did my postdoc in the UK, and I still keep maintaining good relationship with my former mentor and other labs there. And when I need, I don't know, special, for instance, electrophysiology measurement, the letter of collaboration always, I think it really helped that I have some collaborators outside of Czech Republic to help me to address the critical question or the general hypothesis of my project.
Perfect. Thank you for sharing the tips. And Aleš, you have another insights into successful applications from Gutcher. So could you share some tips how you in Gutcher recognize successful gotcha project.
You know, being a reviewer for the gotcha grants, you have to realize that you really are supposed to go through 10 or 15 or even 20 grant proposals.
Wow.
So you don't really, even though you would love to do so, you don't have time to spend one day on each. So the successful grant proposal for me is the one that I start reading the abstract, and after finishing reading the abstract, I understand what's the general hypothesis, what Tom stated, and what are the ways of addressing the key elements of the general hypothesis, and understanding the ways of doing it. And if it does not really happen, your optimism for the grant proposal goes down significantly. So I really advise everybody to structure the grant proposal going from the very simple explanation of where you are heading, and then maybe providing more and more details as the proposal goes on. As you may expect that, the panel at the grant agency has 10, 15 members, and you can hardly imagine that every single member would understand every single proposal, scientifically speaking. So you really have to provide, every single level of the detail to the reader that has no clue what you are talking about, down to the expert that understands and knows the topic. So you should allow every single level of understanding to be read by somebody.
So if you go from, let's say, general picture to details,
exactly,
this is
exactly.
the,
Very often I have grant proposals that are from neuroscience, so I know nothing about neuroscience except for
those are usually excellent grants.
Indeed, they are. And I always view these proposals that way, as I say. But sometimes it's so ununderstandable that you don't know what to do. So I even go to colleagues who are expert in this particular area, and even though you are not supposed to do so, I at least blindly ask them, what do you think, would that be reasonable to study? And he tell me.
Yeah. So even the reviewer has to ask. We had a question from the audience. It was something about how to make sure that you are solving real world problems with basic science. So is it an important part of your research?
Well, the answer will be you'll never know what basic science will bring. So it's you cannot tell how it maybe it will happen that in 10, 20 years it might have some impact, or maybe it will have some impact immediately. You never know. It's difficult to answer.
Is there a way how to guide your let's say strategy or hypothesis
towards applicable
towards real life problems?
Towards real life problems, there's always, or not always, but very often behind grant proposal there are patients. So that could be. But the question is whether if this super rare disease, one of, I don't know, several millions, if it's that interesting for society, it should be, according to my opinion, but somebody might have different opinion. Or the pharmaceutical companies will have very different opinion on this. So that, the major driving force for application are the patients behind the application.
Yeah. I would say blindness is a kind of real-life problem, so you try to target. Here in med school, we have let's say, the opportunity or the advantage that working with clinicians really brings the real-life problems. So how do you connect with your clinical partners? How do you search them, and how do you cooperate? Aleš, Tomáš, this is both question.
Well, we did not too much. But then, as I'm approaching the end of my active, professional life, I feel like I should do something that would really help to somebody in real words. So we actively search what would be the area where we could apply our basic knowledge or the knowledge in the area of stem and progenitor cells. So we ended up, looking to the skeletal diseases, found a partner, and now it took us 3 or 4 years to establish. I think that it is a very fruitful partnership with the clinicians at the orthopedy department, and we really extracted a maximum what we could. And I finally understand what are the real problems that deserve biomedical research and not only the basic one. So it's extremely important, and I keep telling everybody at my department, go and do it.
So it makes sense to connect with
Extreme
clinical
sense.
because
Actually, in my case, it was vice versa. Clinicians contacted me because they knew that I have some specific model to model some diseases, and clinicians contacted me. Hey, we have this weird family or family with very rare mutations, and would you like to make induced pluripotent stem cells for us, and then organoids? So I said, yes, okay, why not? Let's try if this collaboration works, and it worked actually. So it turned out to be gutture application, successful gutture application in a year or two.
So it takes time.
Yeah, it takes time.
And it's about the consistency of gradual talks between you and clinicians. What can be done? What's needed, and it should be a kind of stimulation for.
You really have to do that very actively. Yeah, otherwise, you may live in dreams, as we did. Myself, I did live in dreams before learning what the real clinical medicine requirement is about.
Yeah. That's also my experience from my let's say internship. That there was a lot of lab people, and they were working on something that was almost irrelevant from the clinical point of view. So the advantage of med school is that you can have the list of unsolved clinical problems, and what works for me from the point of clinician is to know what models and what technologies are available here. Yeah, what you are working on. So this is one of the let's say motivations. Why do we talk about science during breakfast? Another point, what do you think is something that holds us back from the, let's say, international excellence? That's something that we here in Masaryk University strive for. So what are the barriers for excellence?
You know my answer. It's administrative workload, definitely. During my postdoc in the UK, I never signed any documents. Everything was electronically. I didn't even took care of invoices.
You don't like paper.
It went automatically to the economic department. There was simple application in only electronic way. I filled what I need. Then the money where the sky was limit, so there was unlimited budget, no limitations basically, and it was just had to be approved by my supervisor, and then in a couple of days the primers or antibodies arrived, and I could start to work. That was really perfect.
Yeah. So the admin load and electronization is something that's really something.
I wanted to just add one thing, that especially for young investigator who has no money for administrative staff, it's really, there's an increasing number of administrative workload. And for me, maybe I will start in sooner, like lowering number of students, because I can't, mentoring them, so many students, so I need really in the near future maybe that's something that really Faculty of Medicine could do something about it and try to lower or go against the flow, let's say, and try to lower, because this is really really difficult.
Yeah, that's something that we will definitely try to address. However, the admin load is something that's related to deposition. Yeah, when you are a PhD student and postdoc, the admin load is and responsibility is something else that when you are head of department or PM of research group. So definitely the admin requirements goes up. But if it's only about admin load, so that's quite okay.
Of course, not only, but maybe Aleš could add something. I will think about.
Yeah, is it about money? Is it about technology that we don't have? Is it about, let's say, the manpower that works on a topic that can really
I think
stand out?
It's not about equipment. We have even better equipment than in UK. It's not about that much about money, I would say. It's really about people, because I experienced recently that postdocs are going to from academia to industry because they have higher salaries and more, let's say, stable position. So yes, the workforce is really could
so
be
the
somehow
critical
improved,
mass.
but I don't know the way how to improve. If it's only salary or there is something else, like higher stability, or because your contract is always up to the 1 year and then it is extended for a couple of years. Maybe this could be also somehow improved, but I don't know how.
Yeah, so about the research, let's say, environment
this
here.
is something behind my reach. So
HR, yeah, thank you. Aleš, what do you think about that? What hinders us?
Well,
is it
I went through all the periods you name. So I went through the periods when we were supposed to order chemicals a year before you needed them. That was before the Velvet Revolution. So you needed acrylamide. You ordered them in June 86. You got it in June 87. So this definitely is not reality anymore. So I'm happy. Then we went through the period when we had no money and we were asked to produce a data. So it's not true anymore. So then we went through a period, then we had the money, had no time to do anything before we were loaded by plethora of different activities to keep us surviving. It's not the case anymore. We do have money, we do even have a time. But, every single period in your life has some, pros and cons. Fortunately, I feel that this period we live now has many more pros than those cons. And this is what I think it's important to realize.
That's why
we have the instruments, we have the money, we have the support by the faculty. I feel that we do.
Well,
maybe I have just a comment, because you asked what can be done to improve. I think these niches like this, making the interconnection in between the clinics, as you said. From the clinic, it would be perfect because the clinicians are experiencing something. We are trying to doing some basic research on something which actually no one cares about. And if you have this platform, from clinician to basic scientists, and then in the scientific community in general, that, Tomáš is doing something and he has this machine or he has this platform and I have completely other project, but I can somehow interconnect. Because as you see, this high-end publication, it's not, sometimes it's like one laboratory which has 20, 30 postdocs, which is not possible really in the Master University because of the stability, blah, 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 all these things. But
because of the space.
But as we are a lot of small laboratories try to more interconnect, because the one laboratory can do this and the other one can do that, and then you can in general the whole community can work on one project which is interesting, and then you can improve, because as Tomáš said, I mean, you can't be expert on everything. And if you want to go to high-end publications, you have to do collaborations. So I think the platform like this, or also with the clinician, would be something perfect where they can tell us, look, I have this observation all the time, and I have no idea how to handle it. And then you know, this interconnection. So I think
perfect. Great suggestions. So hopefully this will serve the purpose. Yeah. You touched on something. That there was also a question from the audience. What's a good paper?
Please, before he takes that, and we leave this topic, I think we benefited much about his elaboration of those interesting parts one needs to put into a grant application. Because, of course, just like you've all said, funds are very critical in research, and getting funds helps to progress, helps you to research more efficiently. So I would like to ask, based on his vast experience, what are those organizations that one can apply to seek grants in Czech Republic?
Yeah, the question will be addressed by Jitka Blašková from the project office. Yeah, we have a portfolio of projects for different career stages, so we will definitely point your attention to her.
Right. Yeah? Thank you very much. And please don't forget the document you wanted to share.
Yeah. We won't forget the document.
Thank you.
Yeah, definitely.
I'll scan it and distribute it.
Can you say a few words about what you consider a great paper? Because there was how to write a lot of great papers in short time. Yes.
Do the work.
Do the work. Yeah.
Do the work. That's the only answer.
There are many ways how to look at that highly cited paper or high impact factor, a good journal, etc. But I would say that there should be something about the merit of the paper. So, is it something that's completely new and a breakthrough,
no,
what's it?
My opinion is that maybe how the paper is done, what methods they use, how they approach to the methods, if they put proper controls, if what they are saying is really right or right. You never know if it's right, but if it's correct, let's say, that's the proper word.
Justified.
Just justified, exactly. And if it's hypothesis driven, I don't like papers that they put some omics. There are like 2-3 genes, and then they put something downstream. I like really hypothesis driven. So, for instance, this phenotype, there is some preliminary work pointing to this gene, and then we tested this gene on multiple models, multiple conditions, multiple approaches, and we think that we are right. That's something how I like that. I don't like these multi-omics papers.
Fishing, etc.
Fishing, yes, exactly. Fishing.
Yeah. Aleš, can you?
Well, I think it's already.
I mean, this is it's really critical approach.
Yeah.
This
I hear you, and I at least partly agree with you. On the other hand, those multi-omics papers can be a great source for follow-up research, right? Because if you do a massive multi-omics research, quite honestly, because I tried that, you really cannot expect always answering some very specific question from that paper. It's more like a generator of questions and a resource to go back to. So I would like to say something on the defense of those kind of approaches, because I think these can be also real valuable. And what important thing to note is, if like the phone book of genes stays as a phone book no one ever opens, then I totally agree.
Yeah, exactly.
That was a bad paper and wasted opportunity, right? But then you can have a paper, okay, I published something years ago, and I had multiple PIs saying, oh, it's such a great resource for us. I publish 6 papers based on your paper because I pick a novel gene which was never connected with something before, and your multi-omics research actually established that connection, and I developed it some further. right?
Yes, I do agree with you, but there are these papers and the other papers, they perform clearly phishing.
Oh, yeah,
we have
I
not,
agree.
we have no clue what to do, so we do this, and then fall out certain
And
kinds of
sometimes
gene
it starts like that.
And qPCR confirmed that this gene was
Yeah.
upregulated, and that's it.
Look.
So it's kind
And
of...
even my paper actually started like that, right? There was a new cool technology called cosy, and the head of the lab told me, oh, I really want to use that technology. How can we use it, right? Can we just do something and let's see what happens? I said, okay, my God, I really hate that, but let's give it a thought, right? And so then we combined it with multiple other techniques, and then it kind of made sense. But the initial idea was, let's just give it a try because we have this cool technology.
Okay.
No, but I agree that many papers, let's say, okay, we have this microarray machine sitting here catching dust, and we have some spare kits. Let's use it without much of a thought. Yeah,
Yeah.
you're right. Yeah.
Yeah.
But the general answer for how to recognize good paper, there's no general answer, I would say. That's clearly based on personal opinion. It's not general rule, I would say.
Yeah.
That's why actually the scoring of science, of Czech science, really suffers because of you can recognize.
Yeah. That's where I'm heading. Yeah, because there is a movement in the evaluation of science. Forget the bibliography, impact factor, citation, quotes, etc. But go to the merit of the paper. Yeah? And the British system is that every group or department just submits 2 best papers, and the reviewers are scoring this. So therefore, I ask, what would you consider a great paper? When we get there, what papers should we submit? No. Well, the time is running fast, so I would like ask the last question. How to go through from the position of a PhD or postdoc to an independent researcher like a research group leader or PI? What's required to do the qualitative jump?
To be in my case, or
I guess
well,
you did it, and
my career path
you're
was
much closer to that step than I am.
My career path was quite a bit more straightforward than Aleš, because after my PhD, which was 12 years ago, yes, I signed a contract for a 5-year postdoc position in the UK, which is very unusual even in these days. However, due to family reasons and also due to project that was not, let's say, fit to my need, because we work for industry basically. It was academic, it was in university, but the project was focused on industry. So I left after a year. I applied for many positions here in Brno. I said I received positive from each other, but I went to my original place, to Aleš Hampl group, or that time it was the Department of Biology or Histology.
No, it was
2013.
Histology. Yeah.
So I went there, and then I applied for a Gotcha grant. I failed, and the next year I received the young version of Gotcha.
Young research.
And then I continue in a gray zone for a long time at the Faculty of Medicine. So in terms of that, I had PhD students, I had other students, I had my own funding, but I had never ever applied for a research group, and I established my research group actually in December. So it's been 3 months. but unofficially, my research group is, I don't know, let's say 9 years or something, 8 years maybe.
Yeah.
But officially, yeah, we have a research group established.
This is a new concept here in med school. So, but the question goes to Aleš. You started to organize your department based on individual researchers. So how do you recognize someone who's got the potential to go there? Or is it anyone gets the chance, and when you got, let's say, some output, scientific output or funding or etc., got the lab or
not really. I'm not really, fan of scientometry in general. So I always try to look at the person as a, person who has ideas, who has a real will to find a novel reality in biology. So I'm looking for a person who is compatible with the community at the department and the faculty. So there many other aspects beyond the previous scientific output that makes me to decide whether or not this particular person is the proper one for us to have.
Yeah, that's great. So this is merit-based, not just...
Exactly,
yeah.
exactly. I lived in the years where there was no scientometry. There were journals. Everybody knew that these are good journals. There were no impact factors, as they were not needed for anything. And this is how it should look like.
Yeah, we are getting back to this, this area. So, thank you. The time's up, time's over, so I think that I have to thank you that you went through the experiment. I was watching the audience. I think that they didn't die from boredom. And thank you for the interaction. And I would like to ask Kitka to finish us.
Yeah, thank you very much. And before we skip to another topic, the gender dimension in the research, are there any questions regarding the discussion between Tomáš, Aleš, and Tomáš in the audience? No? Perfect. So I would like to say a few words about the gender dimension in the research because with the colleagues from the grant office, we went through in past 2 years, let's say, through the evaluation reports, not only of the Gotcha grants, but the European or national one. And we have learned that the gender dimension is a very interesting topic for the evaluators. And we have prepared the web page application and information there that can help you with preparation of your part of your proposal. And we have prepared some short leaflets that can be helpful with some questions that can navigate you how to write and how to describe the gender dimension of your research. So feel free to have the leaflet and to see the web application. And we, from the grant office and with the colleagues from the Department of Research and HR, we are ready to discuss not only this topic and to help you with your proposals and to help you to enhance the number of points that you receive from the evaluators.
Yeah. Thank you, Itka, for preparing that. The point is, this is a mandatory part of every single grant project and is getting much more attention than before. And nowadays, it's not enough to state that we got a gender equality plan and an HR award, etc., etc. So you need to go into the details. So this is a guidance how to provide the details that are required by the evaluators. So forget about the pre-processed paragraph that you would copy to every single grant application that you are working on. You need to elaborate in a greater detail. Okay, so thank you very much for coming. I hope that you enjoyed the croissant, cafe, and small talk. So hopefully next time we will see each other again and have another important guest as a continuation of the experiment. Thank you very much. Thank you guys that you come.
Thank you. Thanks a lot for staying with us for even 1 hour.
You passed.